All right, last time I did this I said good afternoon, but now it's good evening. I do want to finish up this part 2 because I'm going to give you biochemistry students an assignment at the end of part 2 that will have to do with summing up everything of this. So let's go for a little bit longer and let's talk about a specific example of signaling, and then we will go to other specific examples when we get back together in person on Monday. So the question here is how do cells sense insulin? Remember that we had insulin at the very beginning of the class where we talked about it's two peptides, it's got a hexameric structure. It's very important because it started off how biotech even works. And so it's very important to understand what happens inside the cell once you add the insulin drug and it gets into the body system and it engages the receptor. And so that's where we're going to end up talking about the biochemistry of sight, smell, and taste. We're going to talk about all of these areas. They involve sensing something, a different first messenger, but the machinery inside the cell, even down to second messengers in some case, and the other proteins that are involved, follow the same principles that we talked about. They're different first receptors, but the inside of the cell is changed in similar ways. And actually, it's for growth and cancer applies here too. There, the cell senses the passage of time as it waits through different stages, and that's why a lot of this research will actually go on at the Fred Hutchinson Cancer Research Center. The signaling that is involved with sensing is also involved with growth. And growth is a very important area. Insulin is also involved with cell growth in a certain way. So basically we are going to go through part 2, and I am going to give you part 2 early. We're going to talk about insulin receptor and receptor enzyme signaling, and that's going to be growth signaling. And then we're going to talk about G protein coupled receptors. And we're not going to talk about kinases in the cell cycle this year because it's just too much. We have to cover chapter 12 in essentially a day, so we're going to go a little fast, and we're not going to include the exact thing about cancer. You can learn about that, or you can even get a job at the Fred Hutch and then come back and tell me about how it works. But basically, okay, so you're looking forward to Thanksgiving and Christmas dinners, and not everyone has turkey. Some people have a meat substitute, and there's this thing called tofurkey. I'm not even sure if this is still a thing anymore. I'm not sure if it's any good, but these people certainly look like they're enjoying it. But the main thing is, whichever way you do it, when you have a big dinner, you're eating a lot of carbs. Your blood sugar, therefore, will increase, and then your body needs to send a signal. It's not really that efficient to say just detect sugar directly. It's actually better to have another system that detects sugar levels and puts out a molecule that says there's a lot of sugar in the blood, and what should you do with it? Insulin is a hormone that goes along with blood sugar, and it tells your cells blood sugar is high, and you should take this sugar and you should turn it into energy. There's other signals that say take the sugar and turn it into storage, turn it into glycogen and things like that. Insulin is take sugar and burn it. And the insulin receptor looks like this on the outside. What really matters for us is it has that red binding site for insulin off there. It's actually a dimer, and so the insulin can bind. It never enters the cell, but it actually binds the extracellular domain, and it causes a conformational change. Some kind of conformational change. I'm not sure we even have it pinned down, but it causes a shift in the outside of the cell, which causes a shift on the inside of the cell, propagates to the blue receptor domains at the bottom, and the receptors are kinases down at the bottom, which means when they change shape, they turn on and they start putting phosphates on everything. Kinases catalyze phosphate group transfer. The most common way to get phosphate groups is to just get them from ATP, because the cell already has a bunch of ATP around. So you take it, you transfer it to different proteins, and then by putting the phosphates on different proteins, you send the signal that moves through the cell. Like in neurons, for example, there's insulin receptors. Sugar is very important to neurons, and the insulin receptor tells neurons to also grow and make new connections. For example, you can see the insulin receptor is present especially on the dendrins, the dendrites, where it's branching out. You see a lot of red insulin receptor on these neurons. So what happens inside the cell when you first get activated? Okay, so the kinase domain on the inside of the cell is activated, and the first thing it does is it actually phosphorylates itself. So the inactive domain looks like on the left, where you have this loop, and there's 3 tyrosines in the loop. When you look on the right, when it becomes activated, it starts phosphorylating those tyrosines. And the 3 phosphorylated tyrosines now have these big negative charges on them. And so the whole loop flips over to the right. And you see that the target protein then binds. It's the little red thing that's coming in on the left of the right side there. And it's coming in to be close to aspartate 1132, which is now exposed, and the target protein can bind, and itself can become phosphorylated. So you turn on the phosphorylation domain. It first phosphorylates itself, it opens up, and then target proteins become available. It's interesting that the target here is a tyrosine residue, but that's the specific target for this specific protein. Okay. So now you get, in this chapter, you get a whole bunch of things like this. And the really important thing is not to get overwhelmed when you see it, but to break it down and to say, okay, what's important here? Now, if you look at this, you have steps that are from 1 to 7 that are very important, and the earlier steps are usually the more important ones. And so you see step 1 is what we just talked about. Insulin binds, and the receptor itself becomes phosphorylated on the inside. Then the protein binds, the red protein that we had on the last thing, and it's an IRS1 is the protein in this pathway. And it binds and it becomes phosphorylated, and once it's phosphorylated, it flies off, it unbinds, it goes to other proteins, and it goes to a bunch of proteins that you see there on the right under step 3. And here you get to a bunch of different proteins. That's a really big protein complex. GREB2, SOS, and RAF1 are exterior to the central protein here. In the middle of all these proteins, you see that green circle that says RAS. And that is the central protein that gets turned on by IRS1. So, you might guess, that is more important than the other ones. IRS1 goes to RAS, and what happens with RAS is it's bound to a GDP. When it's bound to a GDP, it's off. When the IRS1 comes and sort of kicks it in the side, it lets go of the GDP, it binds a GTP, and the GTP will actually take time to be phosphorylated, to be dephosphorylated, to be chopped up. And so the GTP, when it binds, activates the RAS protein. And then when the activated GTP-bound RAS will bind this next protein called RAF1, and then we get to a cascade. Because step 5, RAF1 will start to phosphorylate a whole bunch of MEK proteins, and the MEKs will phosphorylate a whole bunch of ERKs. And there you have the phosphorylation cascade. Each of these can do, more than one protein. They do a number of proteins, and that way the signal gets amplified. You get a bunch of phosphorylated ERKs. Those go in the nucleus and they do some stuff. As a biochemist, I'm not as interested in the nucleus as I am in the exterior of the cell. As far as I'm concerned, the ERK proteins go into the nucleus, they phosphorylate other proteins, and they end up making the proteins that are the set of genes that are involved in cell division. So the main thing is, when you break this down, you got to make it simpler for yourself. First of all, the green phosphorylation sends a signal. That is true at many of these steps. Then GTP binding means the signal is turned on and propagated through the RAS protein. Gradually, the RAS contains a slow GTP cleaving group. So after it's on for a little while, it will then turn its own signal off by cleaving the GTP, going to the inactive form bound to GDP. And then below that you have a phosphorylation cascade. Just notice that most of these proteins in the cascade are themselves kinases, which means each one, when it gets phosphorylated, it's going to turn around and phosphorylate a bunch more. So it's a little bit like a pyramid scheme, except this one actually works because each of these can phosphorylate many proteins, and you end up with phosphorylated proteins going throughout the whole cell. So the central protein here is the RAS kinase. It binds GTP, therefore we call it a G protein. It is a interior of the cell G protein, although you'll notice that it's actually membrane bound. The little thing on this figure, look very closely at it. It's actually got a lipid anchor, and it's anchored into the interior of the protein of the cell. And the kinase activates the cell, and it includes cell division in most of the cells that it is. It just has to stay on long enough to have a big cascade that goes along, and then all those kinases go and turn on the genes. So RAS is very important. If RAS gets turned on accidentally, the cell is growing accidentally. And sometimes that can happen with a mutation. So if RAS is constitutively expressed and constitutively activated, then it will actually produce cell growth. That's one way that you can get a tumor. Therefore, they study RAS a lot at the Fred Hutchinson Cancer Research Center. So the thing about RAS is that when GTP is bound, it looks like this, and you have these two switch regions, this threonine and this glycine, that are both close to it. They're both like leaning in. And basically it has its own self-destruct timer, because when that threonine and glycine are close, they will actually start to, they'll basically eventually switch, they'll eventually swing out and expose that phosphate to be clipped by other residues. And so the switches open up, but it takes time to do it. So it's like a self-destruct timer. The GTP does not immediately hydrolyze, but it stays in place and it keeps RAS on and keeps the phosphorylation cascade going for a little while. And so RAS is a huge target because there's lots of mutations that accumulate. It's central to sending the signal for cell growth. And also, it's really hard to make drugs against. It's pretty smooth. You can't really find a crevice to put a drug into. But you have, so it binds GTP, and you have some experimental drugs. There are 3 drugs. The red is the GTP that's shown, and the drugs are somewhere on the protein, and one of them is close to the GTP, the one in the lower right. It has a little green circle, and there's a drug that binds to a cysteine found in most of the cancer-associated mutations. So some of the cancers will actually mutate a residue to a cysteine right there, and that's probably one of the things that makes it unable to hydrolyze the GTP, and then it stays on. So let's make something that sticks to that cysteine, and it will stick to the mutated forms of RAS. It's that kind of idea. There are some drugs that prevent the fatty tail from being added. If it's not anchored in the membrane, it doesn't work right. And then there is the one drug that they found that will actually induce a conformational change. Basically, you've got this very slippery but very important protein, and people have tried and tried and tried to find good drugs that will mess it up. And here's one example, the KRAS inhibitor. This was designed by Amgen, and I don't have the details on exactly how it was designed, but just look at that drug. They spent a lot of science to figure out that that particular structure would bind to RAS, and you see that it's actually bound to, let's see where it is. I want to say that this is one of the ones that looks like it binds to cysteine. So this is a covalent drug that binds to the cysteine. The yellow region is where it binds to the cysteine, and the pink region shows there's a histidine that it sort of flips up. So it binds to the cysteine, and it sort of puts its elbow up to change the shape of the RAS, and that will actually shut down the mutant version of the RAS. I believe it's called KRAS G12C. You see that you have a glycine that has been mutated to a cysteine. And so this is one of the example of the green circle on the past thing. So that's the way that insulin works. And there's a lot of other things that have very different outsides, very different binding domains, but very similar insides. If you look at the insides of all these different receptors, they all are blue ovals, which means there's a tyrosine kinase domain inside the cell. But on the outside of the cell, they look totally different, and they bind totally different factors. It's just that the inside works similarly. And so outside the cell, you have these binding of the insulin receptors all the way on the left. Then you have all these GFs. Everything else is GF, which doesn't mean girlfriend. It means growth factor in this case. But you see that these are similar, and they might even activate similar genes and similar machinery inside the cell, because these are all growth factors. That shows you how central this RAS pathway is. So they start in different places, but they end up on RAS. And for example, here's human growth hormone receptor. It looks a lot like the insulin receptor, has that dimeric thing. Growth hormone binds. There's an activation change in the middle, but then you have a different pathway. You have the JAK2 domain inside. So you can also see some similarities in how these receptors work. This is an example of modularity. This is how that works. And so here's, this is a different RAS pathway, but I want you to look at this and you see the outside of the cell. See if you can find the dimeric receptor and see what color it is. There's a dimeric receptor binding like a human growth factor on the last thing. If you look right in the middle on the top, you see the growth receptor. This is a growth factor. The receptor dimerizes, and the tyrosine kinase domains change shape. So then you have GREB2, and you have a bunch of proteins that go over, but then you have RAS stuck to the inside of the domain and made nice bright red so you can't miss it. And you see the little green GTP that is bound to it. When that's bound, then you have a bunch of kinases, and the kinases are shown in the hot pink color. And you see the kinases are spreading out through the cell because that RAS is able to basically phosphorylate, or it's associated with a protein that phosphorylates a bunch of other kinases. And so, and that's how a different pathway works, but these all look the same the way that David Goodsell will paint it. So there's our phosphorylation cascade, amplification. Here's another recent project by David Goodsell. And actually, I just got on Twitter and I found out he actually just did a figure that shows Mycoplasma genitalium, and it shows our adhesin on it. I'll have to show you that to you sometime. But he's my favorite guy. He's, I wonder if I have the book here. I should just take, oh, here it is. Here it is. Because I talk about him so much, one year my students got me this book, so I already have this book. Thank you for thinking of it, if you're thinking of it. But he does a bunch of wonderful pictures about how it goes. And this is one of his pictures. He always starts with the accurate structure, and then he makes it work. Okay, so if you look in here, let's zoom in on this. This is a growth factor binding a domain in green, and it dimerizes. And then you have a bunch of things shown in blue. This time, look for RAS. What color is it now? If you look over, you see the RAS is light blue, and its GTP is hot pink. Once it binds the GTP, it goes over and it binds this RAF protein. There's MEK, there's ERK, there's all the proteins that we were talking about. This looks like the insulin pathway that we just talked about. And again, I'm not going to quiz you on GREB2 or SOS, but I am going to quiz you on RAS. Yeah, and I am going to quiz you on the fact that there's a kinase cascade that happens after RAS is activated. So, this is a lot of complexity. And before we go further, I want you to think about it. I want you to think about the 5 characteristics of signaling pathways and answer the question, why would these 5 pathways go with the kind of complexity that we just showed you in the insulin receptor and other growth factor pathways? In what ways, in this lecture, did you see specificity, amplification, modularity, desensitization, or integration? So I want you to actually write that down, bring it to class on Monday, and we will compare our answers when we do that. Just think of, why does it help to have so many proteins in this? And is it really that many proteins when we talked about the way that it actually works? Why do these pathways take the shape that they do? Well, because it helps these 5 things. How exactly do these pathways promote the 5 things that we're talking about here? So, I will leave you with that, and I'll let you think about that over the weekend. I'll see you on Monday.